Welcome to Ask the Expert, and we're very excited to have Dr. Rachel Bonami and Dr. Dudley McNitt, both from Vanderbilt, talking to us about B cells, and the title of their talk is Sneaking Past the Bouncer, How Anti-Insulin B Lymphocytes Break into the Germinal Center Clubs, which is very funny and also very timely and pertinent. Just a little bit about them. Dr. Rachel Bonami earned her BS from the University of Florida and her PhD from Vanderbilt University. She's currently an assistant professor of medicine in the Division of Rheumatology and Immunology at Vanderbilt and is jointly appointed in the Department of Pathology, Microbiology, and Immunology. Her research program integrates wet lab and computational approaches to understand how B lymphocytes acquire autoantigen specificity and expand in the repertoire to support the development of type 1 diabetes and other autoimmune diseases. Her ultimate goal is to identify specific B lymphocyte changes that track with immune therapy response to provide mechanistic insight into why treatments succeed or fail for individuals with autoimmune diseases such as type 1 diabetes. Dr. Dudley McNitt received his BS from the University of Pittsburgh at Johnstown and his PhD from West Virginia University in microbial pathogenesis and immunology. He currently works as a postdoctoral research fellow in the lab. Dr. Bonami at Vanderbilt University Medical Center, and he's interested in understanding the importance of germinal center interactions in T1D development and how these sites promote changes in B and T lymphocytes towards pro-inflammatory and pro-diabetogenic subsets. His long-term goal is to establish his own independent research laboratory where he can continue his work and identify cell markers and phenotypes linked with type 1 diabetes development to guide the development of future therapies against these subsets. Thank you both very much for joining us, and very curious as to what this talk will hold for us.
So thank you so much for the invitation. We're really excited to be here. And so the first half of the talk I'm going to I'm going to try to give an overview of some of the backstory that led to this recent publication on which Dudley is a first author. And so he's actually going to take you through some of his findings from that paper in the second half of the talk. And we'd love to stop and answer questions at any point in the talk. So I'll try to do my best to keep an eye on the chat, but certainly happy to pause and discuss anything that is of interest as we go. So I think a lot about how the immune system goes sideways to cause type 1 diabetes. And so I have 2 young children at home, so I think a lot about this sort of collaboration that can often lead to destruction, both in that personal setting, but then unfortunately also in the medical setting, which is type 1 diabetes. So there are a lot of early events that are happening sort of behind the scenes that you don't realize. Oh, and I should say no disclosures. So here's me. And so here's a review that I published with Jamie Felton. So she's at Indiana, good colleague of mine. And so this is a pretty simplistic summary of the piece of type 1 diabetes that we think a lot about. So we think lots about these anti-insulin T and B lymphocyte interactions and how this can help us predict disease, but also how this may be leading to disease. And so what happens initially is you have a B cell that can recognize insulin, and not to discount the other islet autoantigens and their importance here, but this is just sort of our wheelhouse, if you will. So we're really zeroed in on the insulin reactive lymphocytes specifically. And so what happens is they can form a B cell receptor that binds insulin, and this allows them to take that protein in, process it, and present it via MHC to a T cell. These CD4 positive T cells can start to become pro-inflammatory. Interferon gamma is one of many cytokines that they can be steered to produce. And this inflammation then can help propagate CD8 T cell destruction of islets. Now, there's actually some evidence to suggest that CD4s can have some cytotoxic function as well, but I wasn't going to get into that today. And so how do we know this whole thing is happening? So in both mice and in people who are at risk for type 1 diabetes, you can find the presence of these circulating insulin autoantibodies that essentially serve as red flags that this communication is happening. We don't fully understand the events that lead to this antibody production, and so that's a little bit of what Dudley will talk about today. Largely, there are 2 main routes by which a B cell can become an antibody secreting cell. One is a T cell dependent route, which is what I just described to you, where they're talking to a T cell and then the B cell is getting activated to become an antibody secreting cell. Alternatively, the B cell can cause problems all by itself, and it can actually respond to innate signals that allow it to go through what's called a T cell independent path to becoming an antibody secreting cell. And so it's, I think, reasonably likely that T dependent pathways are important here. It's, I think, currently not entirely known whether T cell independent pathways are contributing to this antibody that we're finding in people and mice at risk for disease. And so I just wanted to give you a little bit of a flavor of what we do in the lab. So we actually have both human and mouse research programs. I'm not going to talk to you about our human research program today, just given that the paper was mostly, well, entirely focused on our mouse work, but this was largely initiated. So I got into the type 1 diabetes field when I did actually both my graduate and postdoc training with Tom Thomas, who, if any of you have ever met him, he's like the most wonderful human being. And unfortunately, we lost him last year. But so I'm going to talk about our mouse work. But maybe in some future time, we can talk to you about some of the human work that we're doing. So this illustrates this communication between B and T lymphocytes that I was mentioning. So these are actually sections from an entity mouse pancreas, and these are islets. Your islets aren't supposed to look like this. So these are full of lymphocytes and other immune cells. There's some uninfiltrated islet tissue here. So that's what an islet is supposed to look like. It's not supposed to be swarming with immune cells like this. And so on the left-hand side, the brown staining here is the B cell staining. On the right-hand side, same section, but this is the T cell staining. And here are a couple of representative sections, and I think you can appreciate that what can happen is you can form both B cell and T cell zones that are called these tertiary lymphoid structures. And so this was first described a while ago, actually in Peggy Kendall's paper, where she showed that not only can they form these tertiary lymphoid structures, but they can even form germinal centers within these tertiary lymphoid structures. And so an islet is not a place that a germinal center is supposed to form, but that's what we're seeing in type 1 diabetes. So to better understand the antigen-specific B cells and how they're getting stimulated to participate in these TB interactions, especially in the pancreas, one of the models that Tom Thomas had originally developed was this BH125 model. And so the SD stands for site-directed. So in essence, you have an anti-insulin heavy chain that can pair with endogenous light chains. And so some small fraction, usually about 1 to 2% of the B cells bind insulin, whereas most of the B cells in this mouse don't. So a lot of the light chains that pair with that heavy chain do not make an autoreactive B cell receptor, or at least not one that binds insulin. And this model was an advance because now these were isotype switch competent, meaning they had a particular specificity and then they could class switch away from IgM to say IgG or IgA. And so John Williams was a graduate student at the time working with Tom, and Chris was his lab manager, and together they developed and characterized this model, which I'm going to tell you a little bit more about. So Jamie, who I already mentioned, did some work to look at the disease incidence in these animals. And so in a regular entity mouse, there are some insulin binding B cells here, but they're generally not very numerous. And if you try to inhibit them with excess insulin competitor, they're generally not inhibitable. So how specific they are for insulin, we're not sure, but it's clear that some fraction of these cells become antibody secreting cells because you can find insulin autoantibody in these mice. Now, this is a pretty enlarged population. That was the point of adding this BCR transgene in. And so when you give them more anti-insulin B cells, you have a higher percentage of them that develop diabetes a lot more quickly than their entity counterparts that have been a workhorse in the field. So this exacerbated diabetes is something that we wanted to then think a little more about. If we can do something to target this population, what happens? And so I guess I should mention a paper that Tom had published originally that I'm not showing here, where if you, instead of giving them an increased population of anti-insulin B cells, if you prevent the mice from being able to make this specificity, genetically speaking, you get really strong disease protection. So we then decided, this is some work I did with Tom as a postdoc, to try to target these anti-insulin B cells selectively. So when we did this, here's an untreated mouse, and so these are the cells that we'd like to get rid of. And if we injected a second antibody that bound a different epitope of insulin through actually both the FC recognition by other immune cells and also via its cross-linking function in developing B cells, you could eliminate B cells from the repertoire. And so that's shown here. So this is the same type of mouse. It's just when you treat them with the antibody, you get pretty good depletion of those cells. And so if you treat them just for this one specificity, so presumably there are other islet specificities that are contributing to disease, but if you just target insulin, this seems to diminish disease below those that are untreated. And so this is why we're so interested in insulin autoimmunity specifically. And there's a lot of work that I'm not going to go through that points to insulin as one of the major autoantigens in type 1 diabetes. So the other thing that was kind of interesting is that despite having a lot more anti-insulin B cells, if you look at the circulating insulin-specific IgG, that's actually very low, if even detectable, in these mice that have a lot of anti-insulin B cells. Now that seems a little bit counterintuitive because in the NOD mice, we can, as I mentioned, you can find this at least in some of the animals. But what we think is happening, well, not think, what we've shown is happening is that these anti-insulin B cells, even though they're in the periphery and even though they're capable of presenting antigen to T cells, they're blocked at becoming antibody secreting cells. And so one of the immune tolerance mechanisms that's supposed to hold them in check actually is working properly. Unfortunately, preventing autoantibody production is not sufficient to prevent disease because the antibody is not considered to be the major pathogenic effector molecule in type 1 diabetes. It's really this antigen presentation that's the problem. So here's some work that Peggy Kendall had published a while back showing that these anti-insulin B cells can process and present insulin antigen. So these were insulin-specific T cells, and when she co-cultured the B cells with insulin, you can see that she got a pretty nice response from the T cells. In this example, they're producing interferon gamma. And so, whereas if you use a non-insulin specific B cell as that antigen presenting cell, you really don't see anywhere near as much of a response. So there are a couple other models that I just wanted to bring in. So I told you a little bit about this model where we have anti-insulin B cells to track. There's another model, BDC 2.5, where they recognize a chromogranin or also a hybrid insulin peptide, actually is what was found later. And then there's also this 8 of 10 model where you have anti-insulin T cells. And so I wanted to go through a little bit of work that Tom had done with Xiao Wan and Emil Unanue. And in this set of experiments, they looked at these anti-insulin T cells and how they interacted with these anti-insulin B cells. And so what they're showing here is that when you have mice that have both the anti-insulin T cell and B cell, now these B cells are really rocketing off and becoming germinal center B cells. Whereas without the T cells present here, you can see that that's less commonly observed. And if you just have 8 of 10 mice alone, where they have a polyclonal or at least a reasonably polyclonal B cell repertoire, you're not seeing this nearly so dramatically. And so one of the other things they looked at was when you actually transfer in these cells, what happens? And here's what they used in this series of experiments as a control, the BDC 2.5 versus the 8 of 10s that bind insulin. And so no germinal centers, no germinal centers. But then when you look in the pancreatic draining lymph nodes where you now have anti-insulin T cells and anti-insulin B cells, those B cells are starting to do things that we don't want them to do, i.e. adopt a germinal center phenotype. This is definitely true in the pancreatic draining lymph nodes, but you can see this in other sites as well. And then they also looked at the T counterpart, the T follicular helper cell. So these tend to be PD-1 CXCR5 high, and there are quite a few other markers that they express as well. But you can see that before transfer versus after transfer, you start to see this pretty robust population of T follicular helper cells, especially in pancreatic draining lymph nodes, which is a T1D relevant site. And if they added the B cells in with these T cells. So just adding the T cells, the anti-insulin T cells alone can cause some amount of diabetes, which is shown here. But if you add in those B cells, you start to actually exacerbate this further, suggesting that there really is this dynamic interplay between T and B cells that recognize insulin. And so another thing that they looked at, which I just to this day think is just such a beautiful piece of work, they found that it wasn't just any anti-insulin B cell that could communicate in a problematic fashion to a T cell. It was specifically the germinal center anti-insulin B cells that could do this. So in this case, the B cells here are all a given specificity. The difference would be whether or not they have a germinal center phenotype or not. And so 8 of 10 T cells recognize this particular register of insulin that's different than these two T3 cells. And so the 8 of 10s, if you look in the non-germinal center cells, the 8 of 10s are not responding to whole insulin. But when you then substitute the B cell that has a germinal center phenotype, now they're able to respond to this whole insulin that's being presented. And so this suggested to them that there was something unique about anti-insulin germinal center B cells, that their ability, how they processed and presented insulin actually changed when they changed their phenotype to becoming a germinal center B cell as opposed to a non-germinal center B cell. And so lastly, oh, I'm sorry, I actually think I pulled that slide just for time's sake. So then one other thing that got us interested in thinking about, well, how do germinal centers get unleashed when insulin is either an immunogen or autoantigen? And so one of the things you can do is set up these various immunization experiments where you can either look at a T-dependent adjuvant or a T-independent adjuvant. And so in the top here, this T-dependent immunization was using complete foreign adjuvant or CFA. So insulin was mixed in CFA. And then after 3 weeks, insulin-specific antibodies were measured. Alternatively, there's this Brucella ring test antigen, which has classically been shown to induce a T-independent response. And so this is work that John Williams did as part of his PhD with Tom, showing that the T-dependent immune responses. So let's just focus on the VH125s here. These are the mice that have these anti-insulin B cells that I was mentioning. So they really don't make much IgG, either IgG1 or 2A, whereas the wild type mice that were immunized with this same immunogen were able to elicit a response. So there's something different about the anti-insulin B cells that are developing here as opposed to this particular specificity that VH125 generates. Now, if you switch to T-independent immunization, this is where things get interesting. So now they can respond, they make a bit of M, but they actually make some IgG to A or C, depending on which strain you're talking about. This is actually done in the Black 6 model, but we find something similar in the NOD mice, which is what Dudley's going to talk to you about. But in contrast to the T-dependent immunization, where they're really just quiet and there's really not much happening, you can see with T-independent immunization that you now seem to sort of unleash these cells. And these were not just extrafollicular responses. So when John looked, if you immunized just with the adjuvant alone, you see some spontaneous germinal centers that are present. But when he immunized with this T-independent adjuvant, you can see fairly robust germinal center formation anatomically, and also looking with flow cytometry at the phenotype of the cells that were present. He found that there were either unswitched or switched anti-insulin B cells that were adopting this germinal center phenotype. And then here's the same thing, just looking at the number of cells. So this led us to this question of trying to do a better job of understanding what exactly are the signals and the conditions that allow these anti-insulin B cells to enter the germinal center and then become antibody secreting cells. And it's especially this germinal center entry that we would like to therapeutically block, because as I mentioned, we don't generally, there's not a lot of evidence in this field to suggest that the autoantibodies are pathologic directly. So with that, I'm going to transition this over to Dudley McNitt, who's a postdoc in our group. and he's going to talk you through his recent paper that he just published, and I'm going to do my very best to forward the slides on his behalf. So, Dudley, I'm going to mute myself, and I'll turn it over to you.
Thank you, Rachel, and I want to thank Monica and thank everyone for their time and being here. I'm just going to go through some quick highlights of from our paper, productive anti-insulin germinal center B cells, and the stimuli that dictate whether they form or do not form germinal centers. And to get back to Rachel's kind of big picture question of, we're really interested, or I was interested to know what general stimuli will promote germinal center entry for these anti-insulin B cells in the VH125 site-directed mice on the NOD background, and really understand, do they even form germinal centers in the first place? Could part of the reason or the failure for antibody production be due to just poor germinal center responses? So I initially set up an immunization experiment where I immunized the mice in a similar fashion to what Jamie Felton did in her 2018 paper, using insulin, immunodominant peptide, the B chain residues 9 through 23, mixed in with CFA, complete forage adjuvant, but I also added in a control for antigen NPKLH, which normally produces robust germinal center and antibody productions in numerous different mouse models, to just assess, do these mice have an inability to respond to T-dependent immunization with CFA in general? And I immunized the mice subcutaneously at the base of the tail, and followed them for 21 days, collected serum at 21 days, but then I also boosted them at day 7 with the same mixture of antigens mixed in IFA, sorry for the typo, rather than CFA, and then followed them for an additional 7 days to really try to drive any sort of robust germinal centers that could be there, hence the boost. And then at day 7, post-boost, I collected serum to look for insulin and NP-specific antibodies, as well as isolated cells from the draining lymph nodes to characterize the phenotypes of the B cells, whether they are expressing markers associated with germinal centers or as well as something I'll talk about more, apoptosis. So shown here on the left is the anti-insulin, or rather, excuse me, antigen-specific IgG ELISA data, with insulin shown on the left and NP-specific on the right, looking at pre-immunization and post-immunization from these mice, where I found that after immunization, the NPKLH induced a significant increase in total antigen-specific IgG, whereas the NP-specific antibodies were produced. But when looking at insulin-specific antibodies, there was no significant difference and really not a strong increase in anti-insulin antibodies produced after immunization, which is what we had shown previously with Jamie's work in this model. And one thing before Rachel, you highlight that I just want to mention is that we also looked at the type of antibody produced here. I'm sorry I don't show this, but we found that there was none to report for anti-insulins, but the NPs were able to, we found that they produce IgG1 and 2A, so it was promoting subtypes as well. So immunization doesn't drive antibody production, but what about germinal center formation? Could these anti-insulin B cells, are they even making it or forming germinal centers in response to this immunization approach? So this is just representative flow plots to illustrate how we gated or identified these cells, with insulin binding B cells shown here on the left and NP binding B cells shown on the right. And these are gated off of live lymphocytes and looking at CD19 B220 positive cells, then gating for antigen specificity, either insulin shown on the left or NP binding on the right, and then follow that up in subgating base and looking for those cells that are antigen specific that also express the germinal center markers GL7 and FAS, and showing kind of weak or not really a little germinal center formation in the insulin binders, but the NP binders a very high or really kind of strong germinal center response. And when doing and graphing this data, looking at the frequency of germinal center B cells and the antigen-specific gates, as well as the cell numbers, found that the frequency of insulin-binding B cells did not increase after immunization, whereas the NP-binding B cells showed a significant increase in the frequency of these cells and germinal center phenotypes. And this is also represented here in the number of cells were significantly higher in the NP binding, versus a very limited anti-insulin germinal center response after this immunization. So they're not really forming germinal centers after the immunization approach, which led me to question, well, then what's happening to these B cells? Are they not entering, or are they possibly dying in the germinal center responses, as apoptosis is one of the peripheral tolerance mechanisms that controls autoreactive B cells? So I wanted to assess apoptosis levels in these mice in the same immunization strategy. And this is the gating scheme that we used here, looking at insulin binding B cells in the top row, then the middle and bottom rows are NP binding B cells. And again, looking for antigen specificity on the left, germinal center markers here in the middle column. And then on the right, looking at a marker of apoptosis, activated caspase 3 in the insulin binding germinal center positive B cells at the top, NP binding germinal center positive B cells in the middle. And then, as I'll explain more in a moment, a control population we used for normalizing our data, this germinal center negative population, which doesn't express much, if hardly any, activated caspase 3. So when graphing this data, I graphed it based off of the MFI, and I normalized this based off of that non-germinal center B cell population to account for differences in the intensity of the caspase staining over the numerous experiments I conducted. And on the left is a histogram, a representative histogram for the different groups. You can see the total germinal center B cells in the black dotted line, germinal center positive non-antigen binding B cells in the gray. The black solid is NP positive germinal centers, and then the shaded are the insulin binding germinal center positive B cells. And on the right is a graphical representation of all of this data. And we found is that compared to the NP binding and non-antigen binding B cells, the insulin binding B cells had a significant elevation in activated caspase 3 MFI relative to those two groups. And interestingly, when looking at the non-immunized insulin binding B cells, these B cells also in this mouse model had a high degree of caspase 3 expression that was not significantly different from those in the immunized group. So really, the kind of take home for that is that T-dependent immunization with CFA and insulin peptide does not lead to productive germinal centers. And in fact, we would classify these as being abortive germinal centers, where they can maybe form a weak germinal center response, but there's a lot of, or really a higher degree of apoptosis in these germinal centers compared to your more productive germinal centers from a foreign antigen. But as Rachel had highlighted in the beginning of the talk, we have known through the non-autoimmune prone background that T-independent immunization with this insulin BRT conjugate, which the BRT mimics one of the classic type 1 T-independent antigens, this route seems to promote robust germinal center formation and antibody production in the non-autoimmune prone background. So what happens here in the NOD background, and the similar questions of serum, anti-insulin IgG, and flow characterization of the B cells? Here's ELISA data showing that the insulin-specific IgG in either our BRT control population or the anti-insulin BRT conjugate, you can see that the insulin BRT conjugate resulted in a significant increase in the production of anti-insulin IgG relative to pre-immunization, and it was also, but we didn't see the same sort of effect in the BRT only immunized group. And when looking at the germinal center phenotype of the B cells, on the left is the representative flow plots for this of the BRT or the insulin BRT conjugates. You can see, and this is represented in frequency and total cell number on the middle and right, respectively, that immunization in this approach resulted in a significant increase in the frequency of germinal center B cells, anti-insulin germinal center B cells, and in total cell number of this population. So T-independent immunization drives a robust germinal center response. And when looking at the degree of apoptosis in these germinal centers, using the same or rather similar gating strategy that I used for the T-dependent CFA approach, representative histogram on the left, graph on the right, we didn't find any significant difference between the insulin binding B cells and the non-insulin binding B cells, the kind of control population within each mouse, in the degree of apoptosis that was induced by this insulin BRT immunization approach, suggesting that productive germinal centers that form in T-dependent responses don't seem to have this difference in apoptosis expression. And one last little highlight that I want to mention here from this paper is I also looked at differences in caspase 3 expression, so apoptosis marker, in spontaneously forming germinal centers in these mice. And again, the same type of representative histogram on the left, but I looked in two different tissues. Looked in the mesenteric draining lymph nodes, shown here on the left, where Rachel has her cursor. These are lymph nodes that feed or drain out of the gastrointestinal tract. There's a high degree of basal germinal center activity there, and that was a good control for us to look at the setting of insulin binding and non-insulin binding B cells in a tissue that wasn't associated with T1D development. and maybe have a different local milieu and environment. Also looked in the pancreatic draining lymph nodes, a T1D necessary tissue and entity mice, an important site for priming and activating autoreactive B and T cells. And what I found is when looking at the degree of caspase-3 expression in the germinal center B cells in both of these tissues, found that the mesenteric lymph nodes, there's a significant difference in the degree of caspase, being that insulin-binding B cells had a high level of caspase-3 expression in these tissues relative to the non-insulin-binding B cells. And interestingly, that though it trended upward as being higher in the insulin binding versus non-binding, there wasn't a significant difference in caspase-3 expression in the pancreatic draining lymph nodes in these mice. But this data gave us some support to there could be differences in germinal center regulation and apoptosis in these mice that are contributing to the lack of these mice being able to produce insulin autoantibodies spontaneously. So as brief conclusions for our manuscript, and I apologize that one of these points is something we didn't have the time to, I wasn't sure if we had the time to talk about the spontaneous germinal center formation. But one of the things we observed was that in the VH125SD mice, these mice do produce spontaneous anti-insulin germinal centers in multiple tissues, including the pancreas and pancreatic draining lymph nodes. And these, interestingly, despite the fact that they don't make antibody, we do observe signs of class search recombination in these germinal centers, going towards more of an IgG1 phenotype rather than IgG2. And so they can form spontaneously, but the bigger points I found here and showed today is that depending the stimuli, that you can get a different outcome or fate of the anti-insulin germinal B cells in terms of how they participate in the germinal center. T-dependent immunization with CFA and insulin peptide resulted in an abortive germinal center, a limited germinal center formation, increase in apoptosis markers, whereas T-independent resulted in a significant increase in germinal center formation with no change in apoptosis marker expression. And this work is significant because, one, I think it highlights not only what I presented, but Rachel presented, that in this model in particular, it shows that anti-insulin B cells, despite their inability to produce insulin autoantibodies, they can still participate in the disease process due to the fact that these mice develop robust type 1 diabetes. And my work here also provides clues into how insulin-reactive B cells become activated in the germinal centers as to which sort of general stimuli are necessary. And this is important because this can guide strategies that may dictate how we could push these B cells in towards a more abortive or apoptosis driving fate, but a little more mechanistic and detailed characterization is required and warrants for future investigation.
Okay, I can take back over. So I just wanted to thank some of the people who contributed to this work. So we have some older photos and some newer photos just to highlight some of the people whose work I emphasized today. So Peggy Kendall has done a lot of the foundational work, all of which was really started by Tom Thomas. And then here's Jamie. So we talked about some of her work. This is my littlest one going on her very first sailing trip when we went to an AI meeting. And then here's our current group. And I didn't talk as much about, sort of Team Human, as I call it. We really talked about Team Mouse today. And actually, I want to specifically highlight Brian Juice, who did a lot of the work that was in this most recently published paper. And of course, would like to thank our funding, and kudos to Dudley for getting an AI Intersect Fellowship Award about a year ago, maybe I guess 2 years ago. And then, of course, the various core facilities that made this possible. So at this point, we are happy to take any questions that you had, or just can open it up to more philosophical questions, perhaps even beyond the scope that we covered today. So I don't see any questions in the chat yet, but I'm at least seeing the chat. So with that, we'll pause and see if there's any questions or comments.
Great. Congratulations on a really nice paper, and thanks for that presentation. Yeah, if anyone in the audience would like to either raise their hand or unmute themselves or just drop something in the chat, feel free. Okay, people are kind of just hanging out there. I think I would like to ask you guys about, B cells are a little bit of the flavor of the month right now. They're very, people are really excited about the role they have in T1D. There's a bunch of papers that just came out in August in 2023 in Frontiers Immunology with Sue Wong's group and Lee Wen at Yale, novel engineered B cells targeting islet-specific T cells inhibit development of T1D and NOD, and then another from the European group consisting of Sarah Richardson, Sue Wong, and Noel Morgan, among others, in 2022, right, that showing that the gene expression profiling in NOD reveals B cells highly educated in the pancreatic environment during T1D. And then, basically a nice little paper that came out from Eli Lilly Group in San Diego in July of 2023, a non-depleting anti-CD19 antibody impairs B cell function, inhibits autoimmune diseases. How, with all this flood of information that's sort of coming out, where does your model sit, I would say, in sort of the trajectory of the march to T1D? So you've got, this B cell germinal center response happening. How can it play well with these other pieces of information coming out?
Do you want to take that, Dudley, or do you want me to?
I can attempt, and you feel free to clarify or add. Well, I think the ability to track the anti-insulin B cells in this model is really the kind of the big feature or interesting bit for the NOD system. And I mean, we've known that in NOD, if you deplete the B cells, you can prevent type 1 diabetes development. So I would argue that, as Rachel pointed out, we know these B cells, especially in those germinal centers, seem to be helping via the antigen presentation. So I think any of these systems moving forward, and I'm not familiar with the Lee Wen paper, at least I'm somewhat, but not familiar with the other two papers. But I think our model allows the ability to examine what happens with the anti-insulin B cells in the context of, say, this non-depleting antibody, or how antigen presentation is impacted, even when doing in vitro assays and assessing different T cell specificities isolated in the NOD mouse. So I think this is work that is kind of continuation of what we've already been doing. But Rachel, feel free to.
Well, just to kind of think about the big picture, where do we need to go with the field too? So Dudley had a nice summary, and I'll just sort of add a little bit to that. So adults, I'm appointed in the division of rheumatic disease of rheumatology and immunology, and so they treat a lot of people with rheumatic disease, and rituximab is one of the drugs in their in the rheumatologist toolbox. And this is something that adults tolerate pretty well, honestly, better than as an immunologist I might have predicted, but it's not something that you want to give a patient, especially in the middle of a pandemic when you're worried about de novo vaccine responses. And it's even less appealing when you think about treating children who have yet to make the protective memory responses that they need to have lifelong immunity, some of the key pathogens that we all face. And so what we'd really like to do is understand how B cells tick in type 1 diabetes. And how can we be, and I think this is a problem that people are thinking about on the T cell side, of course, as well, but how do you more selectively restrain the immune system so that you can give these therapies, I think are probably going to need, again, not an MD here, but I think these therapies are probably going to need to be given in a more chronic manner to really succeed long term. So right now, a lot of the clinical trials, understandably, have focused on kind of a sledgehammer in terms of the immune depletion or immune function that is being impaired, right? And that's what we have, and that's where we're at. And so they'll give them a slug of whatever the drug is, and then see what happens. Because anti-CD3, rituximab, abatacept, these are not things that you really want to put a kid on for a long time, right? Because you start to worry about the other immune problems that you might provoke. And so if we can do a better job of zeroing in on specific subsets of immune cells, be they B cells or T cells, and just knocking out that function, that starts to be something that you can more easily envision as a more chronic therapy. And where I'm coming from is it looks like on at least the B cell side, if you look at anti-insulin B cells, they're continually emerging from the bone marrow. And so I don't think you can wipe them out once and they're not going to come back. Now, how long does it take them to come back? That may vary person to person. And so there's a reason people can get rituximab once a year, once every 6 months, and have their rheumatic disease controlled. So it's not impossible to think that that might be useful in a sort of reset button, but how quickly do you get back to the same place is sort of one of the problems that I worry about. Now, I also, I'm seeing some questions in the chat here, so let me scroll back up. I'll try to go in order.
Yeah, the first one is from Todd Brusco in
Yeah.
Florida.
Yeah, so he asks, and he's welcome to hop on if he wants to say it, but I can also just repeat it. So he's asking, are there key differences between mice and humans? Probably too many to discuss today, but I can highlight a few. And he says, certainly not as robust of a tertiary lymph node-like structure in human T1D, but some evidence at onset. You know, it's so he honestly could speak to this probably better than I can, but it's you can access human pancreas through the NPOD program that he's very much involved with, but you're still somewhat limited in, you know, how many different individuals you can study and at what stages of disease. And so there's some evidence to suggest that when you have B cells in the islets, that this is an earlier onset. This is often associated with earlier onset of type 1 diabetes, for example, but it's nowhere near, at least what I have seen so far. I haven't seen the tertiary lymphoid structures look anywhere near as robust as what we see in the mice. And so I think there are a lot of, for lack of starting a debate, type 1 diabetes endotypes. I know that's the concept that some people believe in and some people don't. I'm generally in favor of that idea, which is it's not the same immunologic problem that each person has. I sort of liken the NOD mouse to a kid who gets diabetes at age 2, as opposed to somebody who gets type 1 diabetes at age 22. So I think that may be a bit of a different phenomenon in terms of immunologically what's happening. But I think there's a lot we can learn in the mouse system where we have a lot more control, experimentally speaking, you know, in terms of what variables we can test. But all of that, I mean, this is why I mentioned our human piece of the research program, which we didn't talk about today, but that's why I think that's really important. Our hope is that we can take what we learn in the mouse fairly quickly onto the human side and start to think about what questions can we ask with some of the unique repositories that we've built. For example, NPOD, we're not actually doing much with that yet, but we really ought to be. But we do have a pretty strong arm of research going where we look at people who are at high risk for disease. So this is through type 1 diabetes TrialNet. These are first degree relatives who are positive for at least 2 islet autoantibodies. And we know from a lot of work that's been done that that means you have a pretty high risk of disease. And so one of the things that we're looking at and thinking a lot about, we didn't talk about this because I didn't want to get into it and it's not published, but we, for example, see some evidence that there are anti-insulin B cells in human beings who are at risk for type 1 diabetes who are serologically negative for insulin autoantibody. So in other words, they're phenocopying what we've seen for a long time in this VH125 mouse, where you have anti-insulin B cells around, they're capable of causing problems, and they're basically flying under the radar because you don't see the antibody production because they're being held in check. So I don't know if that fully answers his question. There's probably lots more we could say to that, but and then let's see, more founder. Yeah, so Pia mentioned that. So just that they're, you know, that you see more B cells in the earlier onset. So are there other questions or just general? I mean, feel free to jump in with a comment rather than a question if anybody would like to.
You said nice word and nice work, and thanks for taking my question. In terms of like just, you know, getting back to the title of this whole talk, you know, do you have any strategies in mind for, you know, keeping them out of the club, keeping them, you know, at home watching a movie?
Dudley, I'll let you decide how much. Yes, and I'll let Dudley decide what he's.
I don't know if Rachel's thinking the same thing I was thinking, so this
Okay,
may be
if you'd like to share that. Yeah.
different. One thing that we had planned to do, and just for sake of time and other reasons, hadn't for this paper, but it's planned, is I would like to tease out the different there's a big synergy between TLR stimulation and being able to overcome the energy of these B cells to drive their ability to proliferate and become activated. And I'm guessing that there's some sort of difference in the TLR stimulation that's driven by either the T dependent versus the T independent. And that might be a clue as to a route in which we can go to promote the two different fates. And so I had hoped to do sort of a panel of different TLR agonists and maybe even blocking in vitro to see if we could tease out which pathways or maybe likely combination that are needed to kind of drive one fate versus the other.
That sounds like a really fun set of experiments.
I only have two hands, but I'm really looking forward to trying to do that at some point.
I hope Dudley's not agitated with me here for spilling the beans a little early, but he's actually working on a paper right now. Hopefully we'll be submitting it soon, which shows that if you target the germinal center via BCL6, so genetically, if you produce non-functional BCL6 and CD4 positive cells, this leads to complete protection against type 1 diabetes. So this is one of the still arguably maybe a bit of a sledgehammer, but perhaps less of a sledgehammer than taking out all of your B cells. You know, this is an example of a more targeted strategy that you might consider. And so that's actually work that we're following up on right now. We're hoping to show that inhibiting BCL6 with small molecule inhibitors will produce similar effects in terms of disease prevention. But it looks like there is something special about knocking out that germinal center communication between B cells and T cells that, at least in a mouse model, is necessary for type 1 diabetes. We're thinking a lot about the human side too, and so that's work. I didn't talk about her work at all, but Lindsay Bass is a really talented grad student in my group, and trying to keep an eye on the clock here. So she is interested in thinking about how does insulin recognition even happen in human anti-insulin B cells. And so she's thinking a lot about what antigens do they recognize. She's got some data, which is also unpublished, to suggest that they recognize antigens beyond just insulin. And so perhaps some of these other antigens may be in part helping to push them. For example, some of them cross-react with LPS. And so she's interested in expanding that panel out and starting to get a better sense of this. And it, you know, for a while we were sort of trying to decide, is this polyreactivity meaningful? And we went to a Keystone a little while back, and they were talking a lot about polyreactivity to some of these sort of irrelevant antigens, even in viral antigen specific B cells that have pretty high affinity for the antigen. And so I am really interested to see where her work goes. We'd like to understand if that's something that changes as people get closer to diagnosis. That's one of the
That's really fascinating. Is she looking at any microbial antigen?
That's what we would like to do. We have not done that yet, but there is a short list of antigens that we're especially interested in. And so what we've done is we've found the B cell receptors and then made them as recombinant monoclonal antibodies so she can test them against, you know, any antigen that we're interested in. And so that's work that I'm just going to give a little shout out to her. That's her dissertation project, is trying to figure out some of the molecular requirements, if you will, for how does the B cell receptor actually gain this capacity to bind insulin in the first place, and what role, if any, does somatic hypermutation play in enhancing that recognition. And so that's where she's at right now too.
Yeah, I mean, you have a lot of really interesting projects going on in your lab and your collaborative labs, and so I would just encourage people to keep a close eye on what's coming out of the laboratory here at Vanderbilt, which is always known as a powerhouse as well for type 1 diabetes. I guess the very last thing we'll say in the interest of time is if you guys had access to a B cell receptor genetic data repository, would that be helpful to you?
I mean, that's something that we're hoping we can help build.
Okay.
So we're working towards having sequences to put into a repository like that. The nice thing with B cells is it's a little easier. Well, I don't know, maybe people wouldn't agree with me here, but I think you can predict with a little more ease what they might be recognizing, and then you can definitely validate what they're recognizing, I think, in a simpler manner than, say, a T cell receptor. So there are methods to do this on the T cell side too, but B cells are a little easier in that we can just express a monoclonal antibody, and that isn't exactly functioning in the same way as a B cell receptor, but pretty close in terms of what epitopes recognized and
little more one and done. Well, the good news is that we're together with our partners at iReceptor and a number of the prominent T cell receptor scientists in the US and Europe. We're building a TCR BCR data repository. And so stay tuned and keep your eyes on it. And, you know, we'd love to have your participation in
absolutely,
our working groups. If you're interested, I'll send you more information. So
absolutely.
thank you both for really walking us through these really highly informative and mechanistic studies. It's great. And I can't wait to see what else comes from your lab. I hope you guys have a great rest of your day.
Thank you. And
Thank
we really
you.
appreciate the opportunity to be here.
We thank you too. Bye-bye.